Good afternoon, everyone. As Claire introduced me, I'm Shyam Tallury. I'm the peanut and Vigna crops curator in Griffin. And first, I want to thank Brian and Claire and Jill for providing me with this opportunity to present some of our peanut wild species regenerations and characterization research. And I also want to acknowledge my colleagues who are listed here on this talk. Miley Mobley is the peanut technician, and Brandon Tonis is a support scientist, and Dr. Mingli Wong is a research geneticist in our unit. The biochemical characterizations I'm going to discuss this afternoon were done in their lab. So just to give a brief background of the materials that I work with, peanut belongs to the genus Arachis. Many of you probably know that one. So the genus has about 83 named or described species, including the cultivated peanut, which is Arachis hypogea. Of the 9 sections, the genus section Arachis is the largest, to which the cultivated peanut belongs. And many of these wild species are diploid, whereas the cultivated peanut is a tetraploid. And there are 4 other tetraploid species in the genus, which are Arachis monticola, which is in section Arachis, and it's cross-compatible with the cultivated peanut. And the other 3 tetraploid species are in a genetically isolated section, which is section Rhizomatosae. Arachis glabrata, Arachis nitida, and Arachis pseudovillosa are those 3 tetraploid species, but they're completely genetically isolated. They don't cross-hybridize with the cultivated species. Additionally, there are 4 aneuploid species also in the genus. In Griffin, we maintain about 536 accessions, actually, active accessions of Arachis wild species. Most of them as seed stocks, but some as live plants, as they reproduce only by rhizomes, and they rarely set any seed. They're all maintained as live plants in the greenhouses and hardly produce any seed. These species display a very large variation, morphological variation for plant habit and peg length and pod and seed sizes and maturity ranging from annuals to perennials, as you'll see on my next slide here. The pictures on your left there are all the different plant habits, and they range from very compact, small leaf, low or a short main stem plants to which are large leafed and sparsely branched, as you see in the second picture there, and some which are profusely branching and vigorous plants. And the last plant there you see could easily be mistaken actually for a grass. It's Arachis guaranitica. It's in the taxonomic section Trierectoides. And right in the middle of this slide here, those are the pegs, actually, what you see, the peanut pegs, and the little dark structures at the tips of those pegs are the peanut pods. So these pegs could range in length from anywhere from a few inches, like 4 or 5 inches, to about 20, 30 inches long, actually. And especially at maturity, they're very fragile. So growing these species in field plots or harvesting them is very tedious or laborious. So we do all our wild species regenerations in the greenhouses, as you'll see in my following slides. So to the right here, in the picture, they're all the different the morphological diversity in the peanut pods and seed sizes. So they vary from very small seeds or pods to in the top picture, in the very last one in the bottom is Arachis villosilicarpa. It has little larger seeds, and that's one of the species, the seeds of which are consumed by the natives in Amazon region, actually. So as I mentioned, the regenerations of these wild species, we do it in the greenhouses, and annually we regenerate about 25 to 50 Arachis species accessions. And what we do is we take about 6 to 12 seeds per accession. They're germinated in peat pods, as you'll see on the left there in the very first picture. And we grow them in the greenhouses. And when the seedlings are about 2 to 3 weeks old, and then we transplant them into different types of containers, as you see in the middle two pictures there. And depending on the species, we use anywhere from 3 to 6 different containers for each accession to get enough seed to process into the collection. And when it's time to harvest, what we do is we flip these containers on top of these, the screen you see on top of the sifter or shaker. And as it sifts and the dirt falls through into the wheelbarrow and the peanuts stay on top of the screen. So for cleaning and packaging to process into the collection. So as with many other programs, one of our major goals in our program is also, in addition to preserving or maintaining these genetic materials, to characterize them for different phenotypic traits, such as the ones I showed you earlier, the plant habit and pod and seed features. And in addition, we also characterize these species for useful agronomic traits, such as mainly for disease resistances. And recently, we initiated some genotyping studies on these wild species to understand the molecular diversity and identify unique accessions, and down the road in the future to find marker and trait associations, actually. So what we do in Griffin is mostly the morphological characterizations on plant and pod and seed features. We capture digital images for uploading into Green Global. We use the peanut descriptors for collecting this data. And we also have the capability or resources for biochemical characterization within our unit. So I'm going to show you in my next couple of slides about the different types of biochemical analysis we did on these wild species. And right before COVID and during COVID, I started some collaborations, especially with university researchers, as well as other USDA collaborators for genotyping of this wild species collection. And also with the disease resistances, mainly the fungal pathogens, which are a major problem in peanut production areas. So what we did is, as I mentioned earlier, these biochemical characterizations, we have the expertise and resources within our unit. So we picked about 209 different accessions of 45 wild species, spanning about 8 of the 9 taxonomic sections. And primarily, we analyzed them for seed quality traits such as 100 seed weight and seed oil content, and fatty acid profiles, and total protein content. So you see in the bottom here the pictures of the NMR for non-destructive estimation of total oil content. And right in the middle is the GC mass spec for fatty acid profiles. And we have a protein analyzer for seed protein analysis. So in my next couple of slides, I'm going to summarize some data that we collected on the different seed quality traits I mentioned. In the top here, in the graph, you see there's a very wide range of variation in terms of the 100 seed weight, actually, among these different wild species accessions, ranging anywhere from 4 grams per 100 seeds in Arachis dardani to about 32 grams in Arachis glandulifera. The mean was about 14 grams for all the 209 species accessions. As many of you know, high content of oleic acid is beneficial for human health, as well as in extending the shelf life of the manufactured products, actually, particularly those oilseed crops. So that's the reason, that's our interest to analyze these wild species to see what's the range of this oleic acid content in these wild species accessions. And again, we found a tremendous amount of variation for different fatty acids, and also within accessions of the same species, we found variation. However, none of these wild species accessions were high oleic. To be considered as a high oleic peanut, the oleic acid content has to be at least 80% or above. So in our study, although we found a large variation ranging from 14% in Triseminata to about 56% in Arachis veigae. On the top is the seed oil content. Again, we found significant variation among these wild species accessions, all the way from 60% to 44%, with the mean of about 52%. And normally in cultivated peanut, the oil content ranges anywhere from about 45 to 50%. So we got some accessions which are a little higher for the total oil. And in the middle graph there, you see the total seed protein content. Again, a lot of variation ranging from 19% to 37% among these accessions, and with significant variation within species accessions also. And as was reported in many other crops, we too saw a strong negative correlation between total percent oil as well as percent protein content among these different accessions. So the main interest for peanut researchers or peanut breeders with the wild species primarily as sources of disease resistances. So we collaborated with other USDA researchers here, Dr. Rebecca Bennett in Stillwater, Oklahoma, and Dr. Alicia Massa at the National Peanut Lab in Dawson, Georgia. So they both evaluated the section Arachis species for leaf spot as well as white mold resistance. And these two articles have been, our research has been recently published in Peanut Science. And there are several accessions which have been found to be highly resistant to both these diseases. In fact, there are some that had resistance to both of them. So those are the candidate wild species accessions, probably of interest to the peanut breeders to use. And right around COVID, actually, I initiated some genotyping studies with collaborators at University of Georgia, Dr. Soraya and David Bertioli. They genotyped about, again, 110 different wild species accessions of section Arachis using the peanut SNP array. And this work is being summarized or being analyzed, actually, the data, to mainly to see the phylogenetic relationship of these species, and also to identify unique species, actually, for further evaluations. And recently, I also started a collaboration with Dr. Josh Clevenger at Hudson Alpha Institute in Huntsville, Alabama, and Dr. Brian Shuffler, USDA-ARS in Stonewall, and Dr. Charles Simpson in Texas AgriLife Research in Tamu, for genome sequence diversity of Arachis species. The goal here is down the road in the future, we want to develop a pan-genome for genus Arachis. So what we're doing is we took representative species accessions of all the 9 different taxonomic sections, and they're being sequenced, actually, at the Hudson Alpha Center. And the data will be analyzed sometime later this fall, and to see what are the genetic similarities or the divergence of these species, actually, because these, as I mentioned in my introductory comments, these 9 different taxonomic sections are very divergent, actually. So we took a representative species accession from each of those, and we're trying to see the molecular or sequence level diversity on these species, eventually to develop a pan-genome of Arachis. I just want to end with this acknowledging the National Peanut Board for funding the field and greenhouse regenerations, and USDA-ARS for funding, as well as for resources and facilities, with UGA Griffin, where we are located, for resources and facilities, and the Plant Genetic Resources Conservation Unit staff for continued support and help with management of the peanut collection. Thank you very much.